Doing great response to the chat box. Thank you so much. Give explanation. Not really good. Okay, so far it's been okay. For asking. Okay, that is time. Thank you, Zaria. Can I get a volunteer to go ahead and read what the complementary strand would be for number 1? Oh, I know that somebody can. I saw a lot of y'all put the right answer in the chat box.
So, do you want me to just read the question or answer it, or both?
Do both.
All right. So complete the following DNA code with a complementary strand, and the correct, the opposite strand would be C A T A T A T A C.
All right, very good. So that's what we're doing with the complementary strands. Remember that A binds with T, C binds with G. These will be very easy points to get on the star, right? Especially when it comes to DNA. So here we've got cat, AT, and AC. All right, pretty simple, right? Are there any questions about this one?
What is that thing in the middle?
That is an AT-AT.
Uh,
Don't you know your Star Wars vehicles?
nope. I've never seen Star Wars.
So CAT was cat, AT-AT was of course the AT-AT, and then AC, like a AC unit, like air conditioning.
Oh, got it. Thanks.
You're welcome. All right, now here's the next one. The next one, actually, let's go back. Who thinks that they have a really good explanation for number 2? I saw really good ones in the chat and some involving enzymes, so a lot of really good responses. Does anybody feel confident enough to say what they put on number 2? Ooh, nobody. Faye, does your oop mean that you want to go or that you don't want to go? Well, I don't want to have to call on somebody because I don't want to like embarrass anybody, but there's so many good ones. How about How about Hugo? Why don't you tell me what yours was?
Can you hear me?
I can hear you.
I don't think it's right though. I just put that it was splitting up the height, like it wasn't splitting up. Well, it was kind of splitting up the hydrogen bonds, and like when they were splitting up, they had to like repair itself or like replication. They had to like reproduce, right, in order to make more DNA.
There you go. Very good. That's exactly what it is. So here's what I put for mine. I said the DNA helix unwinds during replication, which happened shortly before this. The hydrogen bonds between the complementary bases break, and the new nucleotides are added to make a new DNA strand. I was impressed with a lot of your responses that included the exact enzymes that do this, which again, if the star quizzes you over those enzymes, they're going to probably tell you what they do. There's a chance that they might not, but again, like of the 10 or so stars that I've taken, I don't think I've rarely seen any of them tackle the functions of the enzymes. If anything, they ask like what biomolecule they belong to. That's pretty much it. Does anyone want to tell me what biomolecule enzymes belong to? Any takers? Which biomolecule enzymes belong to? I'll give you a hint. It starts with a P, ends with a protein.
I guess.
Go for it.
Is it proteins?
It is. Good job. Okay. Uh-huh. So if you have any questions about replication, please understand that the star is not going to get too in the weeds about the specific process, as long as you understand that DNA is having to be replicated, which means that we're making new DNA molecule out of the original one. Okay, so I wanted to show you this one more time. We've got our bases, we've got our hydrogen bonds between the bases, our sugar phosphate backbone, and that's pretty much everything you need to know about the structure of DNA. Okay? Okay. Somebody to read the objective for me, please.
Yes.
Go for it.
Students will be able to describe the phases of the cell cycle.
Very good. Thank you very much. So, oh, one more time. This was due 2 days ago, actually. If you haven't turned your biomolecule foldable, please get that turned in. Let's talk about the cell cycle. Actually, before we talk about the cell cycle, let's return back to this image. We talked about this a bit yesterday. Does somebody want to quickly summarize what's going on in this image? Go for it, Justine.
The cell has to copy its DNA before it can reproduce.
Very good. That's an excellent answer.
Good
So,
job, Justine.
oh, thank you, Brandon. All right, so what we're seeing here is we're seeing a cell divide, and before the cell divides, the DNA has to replicate, right? If the cell is going to make a new cell, it's got to make more DNA. That way it's not making just a blank cell, right? So that's what we're looking at here when we're looking at the picture. Oh, Forky's got goodies. I'll take this purple thing. Thank you.
You're welcome.
That looks fun. Okay, so we're talking about cell division then, right? Now, we talked about DNA having to make a copy of itself. Now we're going to get into the actual process of the cell making a whole new cell. Thank you, Kaylin, for putting in the objective. Okay, so here are some images of what we're going to get into specifically. So this specifically, actually, before I get into it, does anybody know what we're looking at? Can anybody name the process?
Like the whole thing, like the
Mhm.
phase.
If you know it, sure.
Prophase, metaphase, anaphase, telophase.
And what is the all-encompassing word for that process? It starts with an M. Do you know what it is?
No.
What was that, Angel? I thought I heard it. So it's
Oh,
it's
no.
like what happens when a scientist stubs his toe. He says, "Oh, mitosis," right? So what we're looking at here is cell division. So we've been talking about the DNA having to replicate so that the cell can divide. Here we're looking at the actual division process. So of course the image on the left is real. This is a plant cell undergoing cell division versus the one on the right, which is a diagram showing the actual process of mitosis. Because you're looking at the picture on the left, it happens really, really fast. This is not necessarily as fast as it happens in real life, but it's pretty close. And there's so much going on that we really have to like slow it down so that we can kind of figure out and really understand the process. So if you're looking at the image on the right, this is mitosis. We're going to get into mitosis more next week. I just wanted to kind of show you why we have to look at DNA replication. The reason we have to look at it is because if you notice in that picture on the right, the DNA literally splits in half, right? So the DNA makes a copy of itself, which is replication. Then when the cell gets ready to divide, the DNA literally like splits in half. Okay? But like I said, we'll talk more about mitosis next week. I just wanted to show you why we're learning this, right? Why is replication important? Where does it fit into the cell cycle? And that's what we're looking at today. So if you could for me, go ahead and get your guided practice. It's located in assignments. It's not an independent practice, actually a guided practice. What it is that there is a blank cycle. We're going to talk about the different stages of the cell cycle, and you're going to fill out that chart. Pretty simple, not too crazy complicated. Very good, Faye, mitosis. You did spell it correctly. And very good, Gigi. That's exactly the definition for mitosis. Okay, let me pull up my PowerPoint. Now, the PowerPoint that I used to use for this was like 50 slides long, so I had to cut it way down just to make sure that we only got the important stuff. I went ahead and uploaded it to the files on Teams. So if you go to the general window and files, you'll find it there. I'm probably going to mess with the other half later. For now, I just kind of messed with that first half, talking about cell reproduction. Okay, can I get somebody to read this slide for me?
I
Go
can do
for it,
it.
Casey.
Oh wait, were we supposed to raise our hand?
I mean, I like that when, who does it? Jacoba does that, right? I like that when you'll do it, but it's not necessary for me quite yet. But y'all can go for it. Go for it, Casey.
Mazari can read it. Was that Mazari?
It was.
Yeah, she
I
can.
don't want to talk to Casey.
Oh my lord, not this again. She can read it, Mr. Martinez.
Okay. Asexual reproduction involves a single cell dividing to make two new identical daughter cells.
Very good. Can you keep reading?
Wait, do I read
whole
the full thing?
Yeah.
Okay. Mitosis and binary fusion, right? Fusion
Mm-hmm.
are examples of asexual reproduction. Sexual reproduction involves two cells, egg and sperm, joining to make a new cell.
Thank you. Very good. So like I mentioned the other
Oh.
day, this is going to tie in to sexual reproduction. So right now we're talking about asexual reproduction where there's no egg and sperm quite yet. We'll get to that. For now, like Clint said yesterday, we're talking about clones. The reason this is important is because like I got a really bad habit with like my fingers. I tend to pick at my fingers and sometimes I make them bleed with a hangnail. What happens is when you start to get rid of all that skin, what does your body have to do? Your body has to make new skin cells, right? Now, you don't want them to start thinking, okay, you know what, I'm going to start being a brain cell on this guy's finger, right? That's not good. What you want is you want identical cells to be replaced. So what happens is when you lose those cells, the body has to make new cells, but you want them to be the same. Now, conversely, when you're making kids, you don't want to have clones, right? You want to have unique individual children because that's how you have a rich gene pool. Think back to evolution and how the brown bear and the polar bear, right? The reason they were able to survive and reproduce is because one of them had an advantage over the other one. They had new genes. So you don't want clones. You want those genes to be kind of remixed. So we'll talk about that when we get to what's called meiosis with sexual reproduction. But for now, we're talking about simple clones, just making brand new cells. Okay, so mitosis, asexual reproduction, this is just them making new identical cells. Okay, can I get someone else to read? Casey, would you mind reading this one?
I can read it.
Go for it.
All cells come from pre-existing cells. New cells are produced for growth and to replace damaged or old cells, and this is called mitosis, which is the M phase of the cell cycle.
Very good. So the actual process of cell division is called mitosis, like I said a second ago. Is because your cells aren't immortal, right? Your cells die eventually. And actually, there's an asterisk there because there are some immortal cells, which we'll talk about later. But that first point from the cell theory, right? All cells come from pre-existing cells. That's why you see that cell dividing into two, right? That one cell becomes two cells. All cells come from pre-existing cells. And again, the reason they do this is to replace ones that are old or damaged. Okay. Now, the instructions for making cell parts are encoded in the DNA, so each new cell must get a complete set of the DNA molecule. So like I said, when I was picking my finger, the cells in my fingers have the entire blueprint for that human being, right? Like that's why when you see on those TV shows, they take somebody's hair or a saliva sample, they could hypothetically clone you because each one of your cells has the whole blueprint for who you are. Now, depending on what genes get turned on and off can determine what's made. So for example, when I'm picking at my finger and I need more skin, the skin part of the DNA is what's red and what makes proteins to make that eventual skin cell. It's not the whole thing. You don't need the whole thing. But each cell contains the entire set of your DNA. And you want to make sure that each one of those is the same because if you get something that's different, that's where you get cancer, which we'll talk about here in a second. Okay. Can I get someone to read this? Justine, would you mind reading it?
DNA must be copied or replicated before cell division. Each new cell will then have an identical copy of the DNA. Occurs during the S phase of the cell cycle.
Very good. Thank you, Justine. So you see there's an original strand, the hydrogen bonds break, and then you get a new strand on each side to make two new DNA molecules. And the important thing is that they are identical. So again, we're making clones. We're making identical pieces of DNA. And this is happening during what's called the S phase of the cell cycle. Here we go. There's a parent cell, two daughter cells. So what's happening is you're taking one cell and you're making two cells. They're clones. It's essentially copying its inside and then just making two of them. Okay, so now we have the cell cycle. Dominic, why don't you read this slide?
Yes. Okay. So like I just say the letters and stuff.
Mhm.
Can you hear me?
I can hear you.
Okay. G1, first growth phase. Synthesis phase, right?
Mhm.
G2, second growth phase. Mitosis. C, I can't say that word.
Cytokinesis.
Cytokinesis. The biggest division is between mitosis and interphase.
Very good.
Nice.
Does somebody want to come off mute and tell me what they notice about this diagram? Specifically, there's two different colors. Does anybody think that they understand what's going on with these two different colors in the diagram?
Aren't they dividing?
That's true. What are they showing the difference between?
I have no idea. I cannot.
Takes a little bit of looking. Anybody think they have an idea?
Can I guess?
Go for it, Casey.
I'm just guessing because mitosis is written in pink and interphase is written in blue.
Mhm.
So is it showing the division between mitosis and interphase?
Very good. That's exactly what the diagram is showing.
Oh.
So think of it this way. Think of it as your whole life, right? Now, it might be weird to think about, but maybe possibly someday you're going to have kids, right? You are going to participate in a ritual that results in another person being created. Now, the amount of time you spend doing that in your life is very minimal, right? It's really short. For the most part, you're going to school, you're having fun, going to the park or the movies, whatever, right? You're doing other things. But there's a small portion of your time that's spent reproducing. That's what's happening with the cell as well. So the cell during interphase just living its life, doing its thing, whatever, it's growing, becoming more mature. And then there's that small M phase where it's for reproducing. So the cell doesn't spend a whole lot of time making more of itself. Sometimes it can, like we talk about with cancer. Cancer has like unregulated cell growth, which means that it just kind of grows all the time and it doesn't really follow a lot of patterns. But for the most part, cells grow, they divide, grow, divide, just like that. So let's talk about G1, S, and G2. Like Casey just said a second ago, this makes up interphase. So again, interphase is not a dividing time, it's a time for growth. And that's what the G stands for. The M phase then is cell division or mitosis. And like I said, we'll talk more about mitosis next week because that whole thing is kind of complicated. For now, we're just kind of looking at the whole cell cycle as it is. So, D1. Can I get someone to read this? Go for it, Justine.
G1 primary growth phase, newly divided cells begins growth cells is making organelles.
Mm-hmm. Very good. So G1, like if you notice, it's happening just after mitosis. So the cell just divided. So that means it's going to be kind of small, right? Because that one cell just became two cells. So this newly divided cell starts to grow, which is why that G is there. G means growth. So we talk about those organelles like mitochondria, chloroplast, whatever, right? All those organelles. During this time, the cell is trying to make up for all the stuff that it just lost during division. So it's making new organelles, it's growing. You'll notice that there's also a G1 checkpoint. Now, this cell cycle has a lot of different checkpoints. The reason that it's there is to prevent the cell from doing something it shouldn't be doing. So for example, this one says G1 checkpoint, prevent an abnormal cell from entering S phase. Now this is a big brain question. This takes a lot of mental ability to answer this. Why do you think there is a G1 checkpoint before the S phase? Remember that during the S phase, DNA is copied. Why do you think there's a checkpoint there? Does anybody think they know why?
Because it's in the starting production of a new cell.
Very good. And so why is it important then that the checkpoint be there before DNA copies itself? What do you think would happen if there was something wrong with the DNA?
It helps prevent the abnormal cells from entering the S phase.
That's true. So let's say, for example, that during mitosis, something happened. Who knows? Maybe there was an error. Maybe you were sunbathing for too long. Maybe you, I don't know, Put your cell phone too close to your lap, whatever. There's some kind of radiation messed with your cells. Let's say then that cell then is damaged. So it divides, and now it's about to reproduce its DNA. That DNA is damaged. They're going to start making a copy of something that's damaged, right? Imagine like trying to copy down someone's answers for the test, but they copy down the answers wrong. Then you copy down the answers wrong, and suddenly everybody's got all those same incorrect answers, right? It can be the same thing here with the DNA. If something happens to the DNA, the body wants to make sure that there are checkpoints in place that will prevent that cell from going off and doing something crazy. Odds are, if that cell is broken or damaged, it's going to lead to cancer. Not always, but most times. So there's checkpoints in place to make sure that the body doesn't get cancer like all the time. It might be weird to think about, but all of light is radiation. So like if you're in a room right now, like we are with these UV lights, these are giving a certain amount of radiation to our skin. The clothes that we wear protect us for most of it. But like outside, we're exposed to the sun radiation, and here we're exposed to light radiation. All these things can be messing with our DNA. So hypothetically, we could be having all sorts of cancers all the time. But because we have these checkpoints built into the cell cycle, it keeps a lot of those things from happening. Now, if that were true and it were perfect, nobody would ever have cancer, right? So clearly there's going to be things that don't work all the time. Otherwise, everybody would be healthy all the time. So there can be failures and there can be checkpoints that are missed, in which case cancer starts to grow and spread and do crazy things. But there is a system in place inside your body that helps you from getting cancer. And those are the checkpoints. Okay, the S phase. Can I get someone to read this one? Whoops, too fast.
Yes.
Go for it.
Sydney is DNA replicated DNA in the cell makes an identical copy of itself.
Very good. So S is for synthesis, and synthesis is basically a fancy word is to make. So the DNA is replicated, right? So they're making more DNA. And again, it's making an identical copy of itself. That's how we pass that checkpoint. Everything is good. The DNA is good to go. We make more of it. Okay, and then now G2, which is the secondary growth phase. Now, this growth phase happens right before mitosis. So we had one after mitosis, right after mitosis, where the cell grows, it doubles its DNA, then it grows one more time to get ready to divide. And here, you'll notice there's a lot more checkpoints. So there's a checkpoint there, there's a checkpoint there. So there's all these other checkpoints just again to make sure everything is good. So there's a checkpoint before it goes into mitosis, before it divides. There's a checkpoint during mitosis to make sure that everything is dividing right. So see, there's all these like things in place to make sure that everything is going according to plan. Okay. Like I said, we'll talk more about mitosis later, as long as you understand that mitosis is the actual process of the cell dividing. That cytokinesis is like the division of the cytoplasm, which I don't think I've ever seen on the star, but it might show up, so I'll put it there anyway. Okay, there is something also called the G0 phase or G0 phase, and that's basically where it just like rest. So after the cell divides and it's like, oof, I'm tired, right? It goes off and just rests for a little bit. That's the G0 phase, which I saw on the star last year, or not last year, but the year before last. So it might show up again. Okay, and then interphase, like I said earlier, where the thing makes more DNA, right? Okay, that's probably enough information for one day. So I think that you've got a good idea of the cell cycle, right? There's a cell, it divides, it grows, it divides, it grows, it divides, right? That is essentially the cell cycle. Next week, we're going to talk more about mitosis, which is that division phase. So when the cell has finished growing, it's copied its DNA, and it's ready to divide. We're going to talk about it next week. I just wanted to really focus on the actual cycle for this week. So let me stop sharing it. So let me know if you have any questions before we move on. Like I said, I don't want to give y'all too much because I know that it's a lot all at once. Avery said, so we get cancer if our checkpoint fails. Exactly, Avery. So like I said, the checkpoints are there to keep us safe, but I mean, cancer is a very tricky thing, and sometimes you can inherit, like when you hear about certain cancers being inheritable, the thing that's happening is that they're inheriting the failed checkpoints, which again, we can talk about a little more in depth later. Oh, wait, not this one. Stop sharing that. this one? Why is it not showing me what I want to see? I think it's trying to tell me that it's this one. Okay, there we go. But I forgot to turn on the sound. Let me turn on the sound. Okay, so you have that. Like I said, it's very, very short. I wanted to make sure that it was short so that you could kind of just get the big points. So the different parts of the cell cycle, and there's like 2 or 3 questions after that. I'm going to give you, let's go 10 minutes. I'll give you 10 minutes to work on that. Let me make sure there's no more questions. No questions. Okay. So yeah, we're going to work on that for about 10 minutes, and then from there, we're going to move on to our exit ticket. Okay.
Sir, can you upload a picture of the cell, like with all the different parts?
Like the cell cycle?
Yes.
Oop, you can't see that. Let me try using the Google. Ooh, there you go. That one's simple. Copy image. Ooh, too big. There you go. Okay, so let's go 10 minutes. Remember, the PowerPoint is loaded into Teams if you need that for support. Yes, GG, sunscreen. Some good answers here in the chat. It's kind of hard to do the PowerPoints and the chat at the same time. If you finish with the notes, GG, you can go ahead and get started on the exit ticket. That's fine. Okay, if you did the exit ticket already, you can move on to your CK through 12. The reading, if you need to, I can post a link for it in the chat box as well. In files, go to the general window, files, and then unit 3. Yep, there's your CK through 12. You're welcome. Uh-huh. That's right.
That's right.
Today is West Wing. Oh, I didn't mean cancer cells. Sorry, GT. Good job, Grace. I'm going to go ahead and put the link to the eggs ticket up, just in case you want to start working on it. It's not crazy long, but it is a little bit longer than the ones we've been working on. So if you're still working on your notes, keep working on your notes. If you're ready for the eggs ticket, go ahead and start tackling it. Remember that you can keep taking your eggs ticket until you get 100. I encourage you to take it as many times as you need to get to that 100.
Mr. Martinez,
Uh-huh.
I'm back. What did I miss?
We talked, wait, when did you leave?
Like 2, 3.
We talked about cancer, we talked about
No, I
science. That was it.
I think I just missed exit ticket.
Yes. Ultimately stuff.